With Lasergene 10, you can assemble your RNA-Seq data using SeqMan NGen, and then view and analyze the results using SeqMan Pro and ArrayStar. To get this set up in SeqMan NGen, use the settings for a templated transcriptome assembly. Then name your project and choose your project folder. Next, you need to load your template sequences. For this example, I'm going to use the human genome package from the DNASTAR website. Now select the read technology and load your sequence reads. Here I have paired end Illumina reads for multiple tissue samples. You do have the option to load multiple samples into one assembly, but for this example, I'm going to run each sample as a separate assembly. This will allow me to load my assembly into ArrayStar for downstream analysis. So we'll start by assembling the brain tissue samples, and then come back and assemble the data from the other samples using the same setup. And I'm going to leave the assembly options at their defaults, and then click Assemble. After creating assemblies for the different tissue samples, I can load the RNA-Seq assemblies into ArrayStar to normalize the data and see gene-level expression values for each of the experiments. To do this, select Start RNA-Seq Project. In the Project Setup wizard, click Add SeqMan NGen Project, and then load each assembly. For this example, I'm just going to load the brain tissue assembly and the universal human reference assembly. And then click Next to go to the Setup Preprocessing page. Because we loaded the template package in our assemblies, the same templates are preloaded into ArrayStar for RNA-Seq analysis. And here we can define the feature types from the annotated reference sequences that will be used for gene quantification. I'm going to leave these at their defaults and then click Next. After preprocessing completes, click Finish to open the project in ArrayStar. Once you've loaded your RNA-Seq assemblies in ArrayStar, you can view a scatter plot of the gene expression values in each experiment. From here, you can quickly select differentially expressed genes and perform clustering. You can then view clustering results in the heat map and line graph thumbnails. You can also download and import annotations from the Go consortium to view ontology information for the genes in your project. Then you can use the filter options to create gene sets based on ontology information and expression values. For example, here I've searched for genes with expression values that are 5-fold higher in the brain experiment and contain the ontology term cell proliferation. I can remember these results as a gene set and view the results in the gene table. From the gene table, we can add columns to see expression values in each experiment, raw read count, and other annotations. Now, if we want to see the sequence alignment for any of these results, we can do that in SeqMan Pro. Let's use this bin1 gene as an example. Here I have the brain tissue assembly open in SeqMan Pro, and I've opened the alignment view to the region in chromosome 2 where the bin1 gene is. And here you can see how the reads align to the template sequence. From here, you can also open the strategy view to see a graphical representation of coverage along the gene. And as expected, we see some sharp peaks on the mRNA features. You can also use SeqMan Pro's integrated SNP report to see a list of putative SNPs in the region of interest. If you have further questions about assembling or analyzing RNA-Seq data in Lasergene, or any other questions about our software, please visit our website at dnastar.com or contact us at support@dnastar.com.